So today we are talking about um humoral immunity. So as I'm sure you guys know by now, humoral immunity is the immunity that is secreted by is mediated by secreted antibodies. Antibodies neutralize and eliminate antigens. And different types of antigens require different effector mechanisms mediated by different classes of antibody like IgG, IgM, IgE. So um, B cell activation consists of first recognition, of course, then activation, and then proliferation and differentiation. This is a good picture of exactly what happens. You have your antigen and your resting B cell. Um, you have, remember first, the activated T cell that will come and activate the B cell. And then you get this clonal expansion, which is the exact same B cell that divides. And then it can become a plasma cell, so it's actually actively secreting antibody. But then it can flip a switch and it can be, or isotype switching, it can become an IgG or an IgE or an IgA secreting cell. But then B cells undergo something called affinity maturation. So although it starts cloning, this over time can get more and more specific for that particular antigen. And that's a process called affinity maturation. And then, of course, as the immune system wanes, some B cells can become memory B cells. So then the general response of the next go around is quicker. Okay. So antibodies, responses to protein require T helper support. Remember, this happens in that lymph node. The T cell is activated first, it travels to the B cell zone where it can help activate a B cell. But if an B cell is actually responding to a multivalent antigen, does not require T helper support. This happens the minority of the time to things like polysaccharides and lipids. And an explanation, I don't think you've actually seen a SCID kit yet, or maybe you have, but you can still sometimes pick up some antibody that is not LOMS, even if they have no T cells. And this is why there is this second mechanism. It's a smaller player. But just like when we're looking at how well our antibody response is looking clinically, we look to see if you responded to protein conjugated vaccines like tetanus, but then we also look at your polysaccharide response, that T cell independent response to polysaccharides to see if you can respond that way as well. Okay, and then as I mentioned in the last slide, activated B cells differentiate into antibody secreting plasma cells as well as memory cells, so you have a faster response next time. So heavy chain isotype switching, that's where that switch is flipped to turn from an IgM to either an IgG or IgE or IgA. In affinity maturation, that process by which it can get more and more accurate for a specific antigen, are typically seen in T helper dependent humoral responses to protein antigens. The primary and secondary antibody response to protein antigens will differ qualitatively and quantitatively. So that first, that primary response, which I think we're going to see here, is not as robust and it's a bit slower compared to repeat infection. That secondary antibody response, where you already have a memory B cell, you get a very robust and early response, and sometimes can actually prevent symptoms altogether. There are distinct subsets of B cells that will respond preferentially to different types of antigen. So there are follicular B cells that will respond to proteins, marginosome B cells, which respond to multivalent antigens, as well as B1 B cells that will respond to multivalent antigens. Again, these latter two are the minority compared to the first. That protein response is by leaps and bounds the most important one that we have. So here are the different types of responses. So you have this very important T-dependent isotype switch, high affinity antibody, long-lived plasma cell. You have your follicular B cells that always starts as IgM. You have your antigen, and then along with T cell help in the lymph node, and get this traditional response that we think about. In the spleen and other lymph node organs, you get these marginal zone B cells, which I think we've already heard about once before. And they respond to this redundant pattern in polysaccharides and lipids. And it's T cell independent and mostly secretes these IgM or natural antibodies. Same in your mucosal tissues, peritoneal cavity, you have B1 B cells that are similar to these marginal zone B cells that will still respond to this repeated pattern. So this is the second stimulus, it's actually the pattern as opposed to the T cell help. And same, T cell independent, mainly IgG, and short-lived. So in general, B cell activation is mediated by antigen recognition in the lymphoid tissues of lymph nodes. You get entry mediated by CXCR5. This is one that I would start to remember. CXCR5 is the signal to get to the B cell zone in lymph nodes. Survival is mediated by BAFF and APRIL. I think that is also an important one to know, that BAFF and APRIL are cytokines that are responsible for the survival of B cells. And then receptor activation occurs through both PASI and BCMA. In fact, I'm going to say for board's purposes, this is a very high yield slide right here, particularly because we know you can get a selective IgA deficiency in CVID if you have a TACI mutation. But I think all these little facts are ones that you should start to put on your note cards to study up. This is just one that I pulled down from Nature that shows the importance of BAFF and APRIL and the survival of B cells, signaling directly through PASI and BCMA. So activation is initiated by binding of the antigen to the membrane immunoglobulin molecule, we've seen before. That membrane immunoglobulin is associated with an invariant alpha and beta chain, Ig alpha and Ig beta, like we can see in this picture. So here's what we see on the outside, and this is essentially an IgM, but it is a membrane-bound IgM, and this is the B cell receptor, and it is in close approximation to Ig alpha and Ig beta, which actually hold the ITAM for signaling. So the other thing that can happen is the B cell will signal when two or more receptor molecules are cross-linked by multivalent antigens. So these are like those B1 cells. You have multiple receptors, and when you're starting to cross-link, oh, I take that back. Do you need two signals even in traditional signaling? I think you do. Scratch that. So you can have T cell help to get this going, but you can also have multivalent. So you start signaling, but it's not going to take two if you don't have T cell help. It's going to take many, many, many of them across the cell to start signal that is powerful enough to actually get to where you need to go to induce these transcription factors. Okay, so assume there's cross-linking here, and then you start getting the tyrosine phosphorylation events. In this case, it is SYK that is binding to this phosphorylated ITAM. So this, in general, in B cell, we call it the Lin-SYK signaling system. Remember, JAK-STAT we learned with cytokine signaling. This is Lin-SYK. It is a similar set of phosphorylation events. Here, though, I want to point out that you have BTK. Bruton's tyrosine kinase. So if you do not have Bruton's tyrosine kinase, here's where your signaling pathway is going to shut down and you're not going to be able to signal through. So you're not going to be able to get any B cell activation whatsoever. But just like we learned before, through these phosphorylation events, you get PLC gamma activation, which can increase calcium, which can lead to transcription factors. You can liberate DAG, which can start that pathway, or you have a similar RAS-RAC or JUMP pathway. All of these can signal these essential transcription factors, which are not different in FET and kappa B and AP1. It's just these early stages that are unique to all the signaling processes that we've learned so far. B cells Lin sync. Commit that to memory, and then remember BTK is on here too. BTK is gone, Bruton's tyrosine kinase. No B cells, then you have Bruton's disease. Get a rest. Because that signal won't work. Okay, there is also a CR2 CD21 co-receptor. So recognize CD21 when we check flow cytometry to look at the health of our cells, checking CD21 often. It's also known as complement receptor 2. So B cell activation is enhanced by signals provided by complement proteins. But I think we've talked about complement before, but remember C3 is cleaved frequently and C3b can bind to antigen, become C3d, and then it's this C3d that can bind to complement receptor 2. So then you have CR2, CD19, and CD81, which becomes a co-receptor complex. I am highlighting that CD21, since this has clinical value, and remember on the boards, all these little facts are now being tested with some sort of clinical correlation. But CD21 is the EBV receptor. It's a receptor that EBV utilizes to do its damage. Okay. So here is that complex that we're talking about. So you have your B cell receptor, essentially the IgM, but then you can have this co-receptor, which is your complement receptor 2 or CD21, complex with CD19, and then the bound complement, which is already opsonized, this microbe. And this can enhance the signaling. On the right here, it's showing a second pathway that can also happen through toll-like receptors, which we've talked about before. If you get toll-like receptor signaling, this is another way to kind of enhance the signal, which would happen through a, it's pattern recognition. So it's patterns. So again, we're thinking about those polysaccharides and those lipids, those repeated patterns you can get. Okay. So when you get this cross-linking, I guess here is the initial response that can happen, all the different things that can start that will affect the B cells as they change. So you have your naive B cell, that means it's naive, it hasn't seen an antigen yet. As soon as it sees an antigen, you get some activation, so you get increased survival, you get proliferation, you increase B7, and then you increase certain cytokine receptors that are important for survival as well. And then you get increased expression of CCR7 and remember a CXCR5 signals to go to the B cell zone, but once this naive B cell has been activated, it wants to go to the T cell zone where it can see a T cell, and it actually can have that T dependent response. So it's going to decrease CXCR5 and it's going to increase CCR7, which causes migration to the T cell area of that zone. Because of all these signals now, it's primed and ready to get the signal from the T cell. So an antibody response to protein antigen requires recognition by both the T helper cell and the B cell. Remember, if you have skin and you have no T cell response, you're not going to get a B cell response at all. T cell stimulation will allow for clonal expansion, isotype switching, affinity maturation, and then eventually differentiation. Okay. So here is this B cell follicle where you're starting to get a B cell that is going to independently first see a part of this antigen, and it is going to upregulate that CCR7, cytokines, B7. It's going to travel with CCR7 closer to this B cell zone. The dendritic cell, remember, is already activated the helper T cell, and then this is the vital interaction that will allow for B cells to then become plasma cells and effector cells. Sorry, I'm getting that. You guys are probably getting the same things. Okay, so if you have B cell receptor recognition of antigen in the B cell zone in those follicles, not only are you going to get those increased in all the co-receptors that are needed for this interaction, but you're also going to get engulfment of this antigen so that it can be presented on a class 2 MHC. And that is the signal that is directly signals to the T cell. So the T cell and the B cell know they're both the right one to respond to this antigen. So the T cells that are going to be activated have already responded to this exact same antigen, bacteria, whatever it is, this protein. And it's going to be traveling towards the B cell zone. And then the activated B cells are going to be traveling to the T cell zone. But it's through this MHC presentation again. So it's already been presented through MHC through the dendritic cell, but it's the second one where it's going to happen with the same MHC class 2 peptide complex. And that is going to fully stimulate these B cells. Okay. So here is a picture of the whole process up close. So you have the antigen, the protein antigen specifically being recognized by the IgM, can engulf, and then it will display it on MHC class 2. And then you get the T cell receptor that is responding to this exact same peptide or has the capability of responding, recognizing the same peptide. At the same time, you're going to start to get co-receptors, and you're going to get a cytokine signal that has already been determined by that dendritic cell. It's going to help and kind of set that milieu that is going to then tell the B cell exactly what it should eventually do, what its fate, and what it needs to be an effector cell. Okay. So after you get this activation, you're immediately going to get B cell proliferation. When this happens, this creates the dark zones because it's very dense in the lymph node. And so it's just a big cluster of B cells altogether. But then it's going to travel, and this is where they're going to start this process of affinity maturation or somatic mutations, which is a similar concept. The two processes, the somatic mutation and affinity maturation, will happen to make these B cells more and more and more specific. But then you can also get isotype switching. And this isotype switching will allow this to become an IgG secreting or IgE secreting plasma cell. But you also get exit of a memory B cell. So we have that response next time. Okay. So those are all the steps of this B cell signaling process. Here is a nice picture of a lymph node. Here's the germinal center. So here's the mantle, the germinal center. Whole thing is the follicle, mantle, germinal center. Here's the dark zone where you're getting proliferation. And then the light zone, this is where you're going to get the isotype switching. Here you can kind of see it too. Here's a darker zone, here's the lighter zone, mantle, and this is all one follicle. This being the germinal center. Okay. So the CD40 ligand CD40 interaction is essential in this process, and we've introduced this concept before. But the activated T cell will express CD40 ligand, and the B cell will express CD40 once these two things have been activated. So the activated T cell will have CD40 ligand, activated B cell will have CD40, and those will interact at the same time you're getting that MHC T cell receptor interaction. And this will stimulate B cell proliferation, differentiation, and germinal center formation. And then it induces something called AID. I do think that's probably an important one to start committing to memory, AID, which will start the process of somatic mutation and isotype switching. So the activated T helper cells will secrete cytokines that, in addition to that CD40 ligand, will stimulate B cells to produce different isotypes. So depending on what cytokine it sees, it will produce different isotypes. Okay, so here are some examples from an experiment that was done. As you can see, if you have TGF beta and IL-5, you get expansion of IgA more preferentially. If you get IL-4, you get both IgG1 and IgE. And if you get interferon gamma, you're more likely to get IgG. The two different subtypes of IgG. Okay, so once again, this is a summary. You get this T cell help to this activated B cell. Well, to partially activated B cell, you get a fully activated B cell, and you're getting a cytokine signal that will allow it, interferon, to switch to IgG, IL-4 to IgE, and then TGF beta to IgA. Okay. So switch recombination is when you get that. Remember, we're going back now to the DNA. And you get that variable region, the joining region. So you get a VDJ region, and it combines with a downstream C region or constant region. And the DNA in between is deleted, and that's how you can isotype switch. So I think we talked about that before. We already get that VDJ rearrangement when the B cell is getting made. And it, by default, will be up against the constant region mu or IgM, because we know the default is IgM. So then when it isotype switches, then it actually has to pop out or delete part of this. It's just like TREX we've seen. Get this recombination. In this example, it's becoming IgE. It's approximating with a constant region epsilon. So you get that same VDJ that's preserved. All the rest of these constant regions are popped out. So it no longer has the capability to turn to an IgG. And here you have a recombination event that will allow it to isotype switch to IgE. Okay, so activation-induced deaminase, or AID, is a DNA deaminase that deaminates cytosines, converting them to uracil. So convert C to U's. I don't think you need to commit that to memory. And then there's something called that pops out the U's. And then there's something called an APE1 endonuclease that cleaves the A basic sites. And this process has been very, very similar to what we already saw in T cell recombination. So as soon as we know we want to switch from mu to a different constant region, aid comes in and it's switching all of these C's to U's. And then comes and it actually pops out all the U's. And then 8 as in the nucleus to actually cause this break. And then you can get a rejoining. And then you have a deleted within the recombined DNA, which will then make it a different isotope. So I think the concept is very similar to what it was in T cells. I would not get bogged down with the specifics, but I would remember aid and are important in this particular type of recombination event and isotope switching. Okay. Affinity maturation is a process that leads to increased affinity of the antibody for a particular antigen. So it's getting more and more and more specific as it is learning. And this is something that is unique to T cells. It occurs only in response to T helper-dependent protein antigens. It occurs in the dark zone of the germinal center. I guess the mechanisms are poorly understood. But we know that there are special dendritic cells called follicular dendritic cells, which are distinct from the traditional dendritic cell that we have already learned about. It is not the same cell. I think the nomenclature is unfortunate. But I think of these almost like a neuron. They're very leggy and they will display different antigens. And then if a B cell recognizes the antigen, it's selected to survive. So the better, more and more it actually recognizes the antigens on these follicular dendritic cells. And more affinity it gains. Those are selected to survive, and the others that are recognizing with lower affinity will start to die off. Okay, so here is an example. You have your variable chain. This is where the affinity maturation is going to happen, not the constant region, which we are just talking about. We're back to this variable region. And you can see here this round ball in the square peg is low affinity. But as you start to get these little mutations that are occurring, this presentation will happen and you will get better, higher affinity. So you're going to get, as time goes on, higher affinity antibody for an individual infection. Part of the reason that you want memory in these B cells, because it's gone under a lot of work to respond to, say, at a strep, for example, or tetanus. You want to keep that around. You don't have to undergo this whole education process all over again. Okay, so here you have your naive B cells. Recognizing antigen is going to travel to the T cell zone. It's going to be a T cell that is going to fully activate that B cell. You're going to induce aid, and you are going to actually get B cells with somatic mutated B regions and varying affinities. Some are going to die off, some are not. You're going to select for the higher affinities. And then only those with the highest affinities after going through this process will be the ones that are signaled to survive. The rest, as they get lower and lower affinity, will die off. Okay. So once this whole process has refined the antibody into what we think will be highly effective against any specific antigen, its next job is to actually become an effector cell, which means differentiation into a plasma cell. So as you can imagine, this also involves major morphologic alterations because it has to start just pumping out immense amounts of antibody. So this is mediated by transcription factors R4 and BLIMP1. It involves the loss of PAX5, will be BCL6. I'm not sure these are highly important. Maybe R4 and BLIMP1 are worth knowing a bit. Change in the immunoglobulin heavy chain gene expression from membrane to secreted form. So up until this point, remember, we just have that part that is connected to the membrane. That's got to go in order to let it be secreted. And that changes in the heavy chain mRNA. Okay, so here we have your RNA. Here is your IgM, mu constant region with your VDJ. But here we need to splice out this transmembrane portion, which will also splice out the cytoplasmic portion. And it gets just replaced by a tail piece that will liberate this and allow secretion. So that's the main alteration that needs to happen to go into a plasma cell. These membrane bound IgMs have then got to go and allow the secreted form to happen. I guess this is just a, let's see. Secret antibody complex with the antigen. This is recognized by the antigen, the B cell immunoglobulin and Fc receptor. And then the phosphatase removes phosphatase B cell receptor complex. This will block signaling. Not sure what this is actually getting at. I'm going to skip this. Sorry. Okay, here is actually probably the most important slide of the entire chapter. Some of the smattering of immune deficiencies that is associated with these processes that we talked about before. Okay, so X-linked AGAM, of course, we've talked about the BTK mutation, but there's also autosomal recessive forms that are probably more likely to show up on the allergy board than even the X-linked version. So this would be like mutations in IgM heavy chains, surrogate light chains, IgA link, for example. Selective IgA deficiency has been recognized with a mutation in TACI. CVID, there's been recognized mutations in ICOS and TACI in some patients. Hyper IgM syndromes, there could be a mutation in CD40 ligand in the X-linked version, but there's also autosomal recessive versions where you can get mutations in CD40 and NEMO. And I'm pretty sure there's one more defect in here. I can't remember off the top of my head. 4040 ligand NEMO. I think there's 4 that will be like lead to. Oh, maybe it's 8 and 9. I guess that's it. These are all hyper IgM syndromes. So if you get a defect in the somatic mutation, isotype switching, you still have IgM. So it's 8 and 9. That's what it is. CD40, CD40 ligand, NEMO, 8, and O. So there's 5 different causes of the hyper IgM syndrome. I don't think these work anymore. Yeah.